We're here live watching as they take an ROV or remote operated vehicle into the depths of the canyon. So far, we have gotten up to, I believe, 9,000 meters, is I think one of the last times we saw it. And what they are doing here, and when I say they, NOAA, the National Oceanic and Atmospheric Administration, along with other agencies such as USGS, researchers, and other scientists, they are using this new technology to help understand this amazing place called the New England Seamount Chain and looking to discover what's right here in our Atlantic backyard. There is still so much to be discovered, and every time they go down, there's a possibility of new species, new discoveries, and new wonders that could lead to better understanding of where we are in the ecosystem and where this ecosystem is. So I would like to introduce you to the Northeast Canyons and Seamounts Marine National Monument. So it is about 130 miles off of Cape Cod, about the length of I-95 going through Massachusetts, and it encompasses 3.1 million acres of marine habitat that, as you can see through the map, there are 3 canyons and 4 seamounts. And all of these are here and uniquely positioned to be biodiverse factories of nutrition and life that thrive right here on our coast. And once again, I am Brittany Peterson, Superintendent of the Northeast Canyons and Seamounts Marine National Monument, working for the U.S. Fish and Wildlife Service, and one of the co-managers of this amazing place. And I would like to go around and have our panel introduce ourselves. And I guess we'll start off with Kathy.
Hi, I'm Kathy Bozak. I am the Fish Passage Coordinator for the North Atlantic Appalachian region of the U.S. Fish and Wildlife Service in our Fish and Aquatic Conservation Program.
And I'm Katie Kubina, and I'm going to be the moderator. I'm here at Mystic Aquarium, and I oversee all of our conservation and research work. And I'm going to turn it over to Peter Oster to introduce himself.
Yeah, hi, I'm Peter Oster. I'm a marine ecologist and senior research scientist at the aquarium, and I'm an emeritus professor in Department of Marine Sciences at UConn.
And I just want to remind people as the moderator that we want to hear from all of you. So go into the live stream chat, join the chat, and while you're learning all this cool stuff and seeing all this cool stuff, put in whatever questions come to mind. And after about 15 minutes, we'll be doing a round of Q&A with our panelists of experts. So we look forward to all your questions. And we're going to go to the ship now to hear from Casey and Kim.
Everyone, thank you so much for joining us today, and thank you so much for having us as a part of this event. So my name is Casey Campbell. I'm the Operations Chief for the Expeditions and Exploration Division of NOAA Ocean Exploration, and with me I have...
Hi everyone, I'm Kimberly Galvez. I'm a marine geologist and the expedition coordinator that's currently training during this expedition.
So we're currently out here on board NOAA Okeanos Explorer. And as you're sitting here watching with us today, you can see the video feed from our remotely operated vehicle, or our ROV. Deep Discover is the view that you're currently looking at. And this is a robot that's about 1.1 miles down on the seafloor right now, or just under 1,800 meters currently. So this ROV is currently exploring Retriever Seamount, which is basically at the furthest edge of the map that Brittany just showed you before of the monument, and just at the edge of the US EEZ. We've been out here on an expedition for the last 28 days, and we are basically working our way back into port in Newport, Rhode Island. But we get the privilege of spending our next 2 days here in the monument. So today's dive so far has been absolutely fantastic. In just a second, Kim's going to tell you a little bit more sort of the context of where we are. And later, Peter will be telling you a little bit more about what we've seen. But we're really excited to have you here today. And I'll kick it over to them to start.
Thanks, Casey. So to give a little bit of context of where we are, over time, these seamounts actually formed via hotspots, and hotspots are actually designed where they're like underwater volcanoes. Some area within the plate tectonics was weaker, allowing magma to actually come through the oceanic crust and form these underwater volcanoes. And through time, it takes potentially millions of years for these volcanoes to actually develop and establish themselves to the point where they're actually considered then seamounts in this area, which is about at least 1,000 meters off the seafloor. Now, over millions of years, we also know that plate tectonics actually shift and glide over the mantle. And this actually promotes more growth of these seamounts areas, where we have multiple seamounts then forming this chain that became the New England seamount chain. And what's known is that these plate tectonics shift about 1.5 centimeters at certain geologic times. They move at around 1.5 centimeters per year, or even sometimes at 1.5 centimeters per million years. So we're going over geologic timeframes here. So as the plate eventually shifts, more of that hotspot actually stays in that center section. And so as the plate shifts, new seamounts form over time. Now we're actually able to see that within the certain range that we're able to collect rock samples from, and we're actually able to age date them, we can tell that based off this trend that we've seen on the New England seamounts chain, that retriever is likely about 120 to 150 million years old. Now this actually is really interesting because certain papers have come out saying that the White Mountains that are within the area of the New Hampshire as well as Maine are actually some of these leftover seamounts, those initially formed seamounts that formed from this hotspot that then shifted with the plate tectonics onto the continental shelf rather than being in the oceanic shelf, as we're seeing here within the monument itself.
So one of the things that's pretty cool about these seamounts is that they tend to host a lot of life and be biodiversity hotspots. So Peter, will you tell us a little bit more about some of the life that we've seen down here?
Sure. So following on from the discussion about seamount formation, these seamounts actually jet up into the ocean. You know, as Kimberly said, over 1,000 meters off the deep abyssal seafloor, and so these become rare islands of hard bottom, hard substrate in an ocean that's generally simply all water, and the seamounts are predisposed to supporting large diversity of animals that feed from the water column, suspension feeders. So that includes corals and sponges and a variety of associated species that can take advantage of the structure of the corals. We were just looking at before from the live feed, there were crinoids that are related to sea stars, the group of echinoderms that are living on the tips of corals, placing them higher up in the water column off the seafloor to be able to capture particles for food. So these seamounts sit in different oceanographic regimes, like we're familiar with the Gulf Stream at the surface. There are also deeper ocean currents that move in different directions. And so the animals living on seamounts are bathed in different water properties, much like plants and animals that live on mountains on land. So there's different life zones as you climb up mountains. There's different life zones as you drop down through the water column and down from the peaks of seamounts and down the sides of submarine canyon walls. So there's just huge diversity of animals that live on the seafloor, over the seafloor, and then because of the currents that drift by, there's interactions with midwater species with those species on the seafloor and provide additional food for those animals. So I mostly work on landscape ecology and how fishes interact with the landscape, both on the seafloor and in the water column. So multiple species of fish, some that we've only seen for the first time live, we've seen on the Seamounts expedition. And being able to work in the monument is special because this is an area that's going to be protected for the long term, and we'll be able to understand how changes in the monument contrast with those outside and across the entire North Atlantic basin.
That's a really good analogy, Peter, by the way, when you look out of the ocean, it's almost like it has a mask over top of it because we don't see these features that are under the water. We just see this reflection and the sky turning back. But with these seamounts, they do, like you described, it acts as a mountain, like we talk about with the White Mountains in New Hampshire, that some of the mountains, as they're taller, they actually create their own weather patterns sometimes. And, you know, similar, not saying this is the exact similar effect, but as you move up, different things change, tree formation changes, and we're seeing the same thing here, only it seems like the top here gets a little bit more diversity on it than we see on the more terrestrial mountains.
Well, I always describe these kinds of expeditions, exploration or research, as climbing mountains from the top down, as opposed to the bottom up like we do on land.
You know, I have a great question that relates specifically to seamounts. We have Anne asking, I think this might be a good one for Kim, being our resident geology expert. Are the seamounts still active once the hot spots move on?
So that is a fantastic question. So once the hot spot is the one that actually stays stationary, and the plate tectonic is the one to move over it. So as the plate moves, eventually the plate will move just far enough where the seamount will actually then be cut off from that hotspot, so that source of magma. So once these seamounts move over that hotspot and are moved away from it, they are not considered any. They're no longer considered active anymore.
Thank you, Kim. And this is a stump the researcher question here. What is the newest species found in this ecosystem? So Peter, I know you've been studying this system for upwards of 30 years, but are you clued into what the newest species is that they've discovered?
Well, so thanks for making me feel old. And well, I guess I would venture an educated guess that it's a coral, and I try to keep. So when we started doing this, I mean, the ecological exploration of these seamounts only began with the very beginning of the ocean exploration program. Myself and colleagues were part of some of the first cruises using Alvin and Hercules, and we thought we were going to do more community ecology, being able to describe who all the players were and how they were related. But ultimately, or maybe not even ultimately, we quickly realized we can't put names on a lot of these beasts that we see. Because the early oceanographic explorations where people dragged dredges over the seafloor, you couldn't drag a dredge, at least with old oceanographic ships, across these seamounts very easily. You'd lose your gear. And so a lot of the work that's happened between 2001 and today has been describing new species using modern methods, like both the structural descriptions and genetics. And so a lot of there continues to be revisions and descriptions of new species of deep sea corals from these seamounts. But this isn't the only group of species where there have been new descriptions, at least across the global ocean. There's been small organisms that live in the seamount sediments, sponges, sea stars, etc. But the newest one certainly I would imagine has been a coral.
So one thing I'll add there, Peter, that people are usually pretty surprised about is just how long it takes to go from the time of collection to the time of a new species designation. So on average, it's about 15 years that it takes to go from that. And it doesn't mean it doesn't happen quicker at times. Just recently, some of our partners worked on species descriptions that got written up in about 2 to 3 years. So it can happen. But on average, it takes so much longer. And that's because there's such an incredible amount of work that goes into it by taxonomists, where they have to research basically every animal that looks like it, that might be associated with it, and clearly make a case amongst their colleagues and the rest of the research community that this is unique, this is new, this is something that's undescribed at this point in time. And that really surprises people, that we can't just look at something and say, oh, that's new. Because a lot of times when we're out here, we're seeing things we've never seen before, and we're seeing things that are surprising or interesting and just different. And that's because so little of the deep sea is actually explored, that every time we're down here, we do see something completely new and different. And it's so exciting. But it's hard to say, like, what's a new species, because you have to do so much of that morphological and taxonomic work and the background studies that have to go into just designating new species. It's part of the reason all the samples we collect go to the Natural History Museum at the Smithsonian, and where they can all sort of live and be used by the community as a whole very shortly after we complete our expeditions.
All right, so a couple.
Can I just add one more thing?
Go ahead, Peter.
Is that I still got 30 seconds, right? I'm kidding. Is that here we are off the northeast coast of the United States, one of the most well-studied parts of the global ocean, and we're still finding big stuff that doesn't have names. You know, as I like to point that out to people, is that all the biological exploration doesn't necessarily happen in rainforests and remote parts of the ocean. It's like here we are in one of the most well-studied parts of the globe and still putting names on big stuff that we're seeing for the first time.
Absolutely.
So a couple of our people who have tuned in are looking for a narration of what we're currently seeing. This initially came in, I think, when we saw that big green, bright green thing and a peach-colored sea star. Oh, I'm seeing the green thing coming up on the screen again, too. So can you tell us specifically about what some of the things that we're seeing right now live, given the ROV's dive in retriever seamount right now?
You want me to do that?
Yeah, Peter, why don't you start and we can add some stuff if you need it.
Sure. Yeah. So well, you can't see the cursor on my screen. So we're at the very bottom of the screen now. We're going over, a sea whip, either in the genus Lepidisis or Keratoisis. The yellow blob is a sponge, Pharia, and the reddish bushy thing to the left of the yellow blob is a black coral. And as we're moving across, there's another sea whip. There's a glass sponge, the little vase like object on the boulder that's kind of in the middle of the right side of the screen. And if you actually looked at any of these if we zoomed in on any of these, you would find other organisms that are associated with the corals or sponges. And these are often, the species are linked to these associates or commensals are linked to the particular species of coral or sponge. So at the beginning of our dive this morning, there was lots of these little parasol or corals. I mean, they look like palm trees. And associated with each is a basket star. And the basket star recruits to the coral when it's young, and they essentially grow old together, like a married couple, as a colleague of mine describes it. And so every once in a while too, I've seen as we're talking these tiny little black fish dart through the camera, and they're called Bristle Mouths. And there are 13, 15 genera in that family, and these are likely the most abundant vertebrates group in the entire world, and they probably have more biomass than any other group of organisms as well globally, and they are important food for a variety of larger predators in seamount ecosystems. I'll shut up now.
No. So what are we looking at here? If they just zoomed in on that cool thing.
Sponge.
Oh, that's a sponge, a glass sponge, and you can so sponges filter, inhale water from the surrounding water column, and then extract nutrients by a very primitive form of digestion. And all the schmutz that you see on the outside of that is the stuff that's caught on the inhalant pores of the sponge. So that's they're very sloppy eaters. And that was a joke. Thank you for laughing. What else? There was a looked like an Acanthogorgia coral there as well. These little
There's been a huge.
Oh, go ahead.
During the course of this dive so far, there's been a huge diversity of coral. We've seen several black corals, those bamboo corals that we were just talking about, sponges. We just saw the little parasols that Peter was just talking about was just in view about 10 seconds ago. Might be off the frame here. It's just one of the things that I think is so impressive about the deep sea, and it's so important when we think about the monument, is that a lot of people think the deep sea isn't really full of life. It's kind of barren, it's void, there's nothing there, out of sight, out of mind. But dives like this, and this seamount in particular, as well as several of the others here in the monument, really show us that's just not the case. There's so much life here, and it's such a biological hotspot that we can learn so much about the deep sea and these habitats. But every day we're gaining new data, and it's helping us to inform what we know about these areas.
And not only that, being able to actually see this footage live via our telepresence, but also sending an ROV down there to take the imagery and the video of these sources, we're not only able to see what is down there, but we're also able to see the behavior of certain animals that are also in this area. So animals that maybe we didn't, or behaviors we didn't see had we been able to pluck it up, or earlier in the days when the only form of sampling or knowing what was down there was trawling. These are all certain behaviors that then we wouldn't have been able to observe. And whether it be a predatory event, via a sea star consuming a coral, or a fish consuming something else, or the infamous swordfish fall before that we had seen a few expeditions ago or a few years ago. It was great to be able to see all of these animals interact with each other, and how the entire environment is reliant upon a lot of these different factors that come in. First, it's the geology that's standardized, and you can see here there's so much of this rock and sediment that creates a lot of that foundation of where these animals will then come and settle upon. A lot of these coral colonies do need a hard substrate to start with their larval in order to actually colonize the area. Then the corals can then bring about more different kinds of species, and then fish, and it's an entire food chain that we didn't know existed even just a few decades ago.
Kim, that's a really good segue to Kathy and her program within the U.S. Fish and Wildlife Service. And with one of the things the U.S. Fish and Wildlife Service does is try to remove the barriers for fish that migrate from freshwater to saltwater and vice versa. But also there is so much mystery, as Kathy can share, like about these creatures that migrate from freshwater to saltwater and back and forth. And yeah, Kathy, can you tell a little bit about like the lamprey work that the Fish and Wildlife Service does and some of the other species?
Sure, thanks, Brittany. Yeah, there's a lot of connections between the conservation work on land and in rivers and the life that we're seeing out in the monument. The Fish and Wildlife Service, NOAA, and our partners are working on conservation across the landscape and the seascape. Because we know that these systems and the species don't recognize political boundaries or are artificial constructs of where one system starts and ends. It's all connected. Species move between areas and sometimes really long distances. And the species and systems are really interconnected. So what happens up in the headwaters of a river is connected to what's happening in the ocean and vice versa. So one really good example of this, like Brittany mentioned, is the migratory fish. Here we're thinking about fish like river herring. That's a group of fish made up of blueback herring and alewife. These are fish that are born in river systems and then spend most of their adult life out in the ocean. And they return to their home rivers to spawn. And then there's the American eel that has a reverse commute. It's born out in the ocean and gets carried by currents into rivers and streams and grows for decades in freshwater, and then treks out through the ocean to the Sargasso Sea to spawn. These migratory fish are really important to freshwater river systems, and they're key to the ocean food webs as well. They're species that are found in river systems across the East Coast, and they're also found out in the monument, where they serve as prey for larger fish, sharks, and whales. Unfortunately, we have seen some of these migratory fish species decline, including one big reason for the decline is the construction of barriers in rivers that keep fish from completing their migrations and reaching important habitat. These barriers include dams and undersized road crossings. So Fish and Wildlife Service and NOAA have several programs that focus on restoring river systems, particularly removing barriers for migratory fish. So we remove old obsolete dams that people aren't using anymore. We make sure that road crossings over streams are designed well so that rivers and fish can pass freely underneath. And in places where we can't remove barriers, we install fishways and fish ladders. So while we're restoring our river systems, we're addressing what's happening across the landscape in terms of land use, pollution, water use. All of these have an impact on our river systems and migratory fish. We also raise fish and aquatic species in our hatcheries to supplement the wild populations and work towards sustainable levels. And we really want to restore these fish populations because they're important to commercial and recreational fisheries, and they're an important component of several ecosystems and food webs, both in the freshwater and the marine side.
Thank you, Kathy. That's a great segue to another question that I have. I think that was really important for people to know, why is this place hundreds of miles off the shore? How is that connected to me and to people and what we're doing on the land? One of our participants is curious about the connectivity between these areas. So the question is from Adam: is there strong community connectivity between the seamounts in the National Monument, or are they distinct and isolated from each other? What are the main drivers of the connectivity effects? Peter, that might be for you.
Sure. So, that's a great question. And the answer is we're still learning about connectivity across the seamount chain and to the continental margin. Some work from some of the earlier expeditions with one species of fan coral found that there were distinct genetic groupings of the corals that were collected across the seamount chain, indicating that there was some isolating mechanism. I'm trying to explain this without using too much jargon. So there were 5 different species of Paramaricia, which is the genus. 5 different clades of Paramaricia, which is the genus. They all look very similar. They're kind of potentially cryptic species, but they had different distributions, suggesting that there was some way to some mechanism that isolated them on different seamounts. There was also some overlap between those clades. So there's some sets of conditions that allow them to have connectivity, and then others that don't. And how all that works remains to be worked out. And it doesn't exactly overlap the patterns that were found for some of the commensal species, that little crabs that live on black corals. They had different patterns. So some's related to the life history of the species in terms of how they are isolated on different seamounts, and some relates to we would infer from the oceanographic patterns that connect the seamounts at different depths. So that might be a little more complex than everybody was hoping for, but it's complicated and we're still figuring it out.
So right now, Peter, on the screen, people are getting a close-up zoom in of something super cool. Can you tell them what that is?
Yeah, so that's a species that's related to king crab from Alaska. So I would suspect the genus is Lithodes. It's got all these little spines, and which suggests at least it evolved under threat of predation, and it feeds on invertebrates on the seafloor.
And can you talk about the 3 things that are around it?
So those are Ophiroids, or brittle stars. So they're sea stars, like you see at the beach. They have a central disc and a mouth, and the arms are thin, and in different conditions, they can either filter seawater for food, that they'll sit on the seafloor and raise their arms and then move food particles to their mouth, or they'll get food particles from the sediments on the seafloor.
Another thing that we saw earlier, and we didn't have time to talk about, but someone put in a question with a great description of it. It says, we just saw what looked like, this is from Judith, we just saw what looked like a rope of flowers attached at one end to what looked like bones with 3 black bands. What are they?
So that was we were looking at a whip coral, a bamboo coral. So bamboo corals have skeletons that have nodes and then a skeletal material in between. And so the black bits were the nodes. and then the rest is, which is more organic, and then the rest is skeleton. And at the tip, that was the live coral tissue. So something had been munching on the coral. I looked closely at that, and it looked like there were some little arms on the other side that we couldn't see. But there are multiple predators that'll feed on corals, like sea spiders or sea stars. I mean, there wasn't at least a large sea star there. So something had been preying on that, and what's left was the coral tissue at the tip, or the upper part, and then at the very tip there was a crinoid or a sea lily that looks like a flower.
Thank you. So we're seeing all this amazing imagery in real time. Tom has a question about the machine, the underwater robot that is providing us with this. And he wants to know if it's tethered or autonomous. And maybe Casey, you can share with us a little bit about the ROV, what a remotely operated vehicle is, and the one that you have aboard the ship, and how it functions.
Sure. So what you're seeing, the video that you're seeing right now is from ROV Deep Discoverer. So Deep Discoverer is a part of a two-bodied system. So technically we have two ROVs, Deep Discoverer and Sirios. Sirios is, if you look at our live video feed on oceanexplorer.noaa.gov, you'll be able to see basically the 3 camera feeds that we send ashore every dive every day. So from D2, which is kind of the view that you're seeing right now, and then Sirios is a tethered system. So they end up basically working in tandem together. And then the ship is connected to Sirios. So Sirios provides guidance, perspective, as well as additional lighting for D2, who gets really, really close and gets to look at every single little small organism that we're seeing. We work with a group of really talented engineers and pilots from the Global Foundation for Ocean Exploration, who basically have built and maintain and then end up piloting all of our vehicles. So when you hear on the live video feed, I know you're listening to us right now and not the video feed, but a lot of times on the video feeds, you'll hear the pilots talking, and they are basically driving the robots as we go. So while we're not down on the seafloor, they're manned from our control room, and they are tethered to our ship. So they're pretty amazing vehicle.
It is, and our audience is being treated now to like this great landscape view with all this neat stuff in it. I think there was a close-up of a very cool sponge. You know, one of the questions that has come in is does light penetrate? Like, how does everything grow down here? Does light penetrate, or how does this ecosystem work?
So we are so far below where light would penetrate. So this is one of the things that I think has been most exciting over the course of deep sea science that we've discovered, these oases in the deep sea, where a lot of life on the rest of life on Earth is really tied to sunlight. And you would think that down in the deep sea, there wouldn't be life. These organisms get particularly the ones we're looking at right now, do end up getting their energy from the water column, as Peter was describing before. So it does end up starting up at the surface and eventually does trickle down the particulates that they're taking from the water column, their food that comes down from the surface of the ocean or from that top photic zone. But they don't have life or light down here. They have life, not light. Unlike a lot of corals that we see in shallow waters where there's a relationship with a photosynthetic algae or a zooxanthellae in their tissue, and they basically grow their skeletons directly from sunlight, that doesn't happen here in the deep sea. They're uniquely adapted to live in a lightless area. So a lot of the colors that we see, and it is so incredibly colorful down in the deep sea, are adaptations that those organisms have, not necessarily for light, but for other reasons. So you might see organisms that are red, like the sea star that we're zooming in on here now, in kind of a pinky color. Well, that color actually disappears in like the deeper down you go in the ocean. So a lot of times we'll see jellyfish with red on them to protect whatever, or they're red to kind of hide any bioluminescent animals that they might eat. So color has a very different meaning down here, but it's really interesting. And I'm pretty sure, Peter, you probably want to jump in and add something about some of the cool fish that have really cool adaptations down at depth as well.
Sure. So we've got the fish we see in these landscapes. Some evolved in shallow water and moved deep. Some evolved in deep water and moved shallow. So some of the deep water animals are well adapted to living in the cold and in low productivity environments. So they don't make a lot of investment in speed. But they do in efficiency. And as Casey was describing, food not only comes from the top down, but it comes horizontally as well. So primary production in other parts of the ocean, upstream in terms of the currents that impinge on the seamounts and in the canyons, is moved by the currents. And so being in an area where there's high flow rates enhances the amount of food delivery. This is why you see a lot of species living on the tops of corals or on the tops of sponges to collect particles that move. But the fish, the high diversity of fishes, some produce their own light. Some are really good at ambushing. They look ferocious. They have big teeth, like the tiny little black fish that I was describing earlier that are called bristle mouths. They have a mouth full of teeth, but they're tiny little fishes, only a few centimeters long, and they feed on plankton and small shrimps. There are animals like gulper eels that their lower jaw is disconnected from their skull, and so they can consume animals nearly as large as they are. I don't know, all I got right now.
Someone was kind enough to answer this question, but I think it's worth repeating for everyone who's tuned in. And people wanted to know, is this feed public? Can I tune into the live feed? Can I hear the audio of the ROV pilots? So I'm going to turn that over to Casey, because I think the answer is yes.
Yes, absolutely. So all of the video footage that you're seeing right now is on our website, oceanexplorer.noaa.gov. You also can follow us on social media, where there's lots of links to our the live video feeds, as well as our website content. So our website has all sorts of videos and imageries that are produced throughout the expedition, as well as summaries of the dives that we've done, and then little mini highlight videos and photos of everything that we've seen. So yes, go check it out. Check out the website. We've got the dive today and then one more dive tomorrow. And then we'll have another expedition offshore the Southeast US this fall.
So that's amazing, because you're able to collect all this new information and samples and video and data. I also wanted to mention that right here at Mystic Aquarium, we have a super cool exhibit on the Northeast Canyons and Seamounts Marine National Monument that we've been developing along with U.S. Fish and Wildlife Service. So I also encourage you to come here, because we have a great rock wall with some critters on it and some amazing video and graphics. So because it's so far away, we wanted to kind of do our best to kind of replicate the Northeast Monument right here at Mystic Aquarium, so that we can kind of bring the monument right to the people. So I think that term has come up a lot, and I just want to make sure we give someone an opportunity, maybe Britton, to talk about, what is the Northeast Canyons and Seamounts Marine National Monument, and why is it this area that's being protected?
Thanks for asking, Katie. Yeah, so the Northeast Canyons and Seamounts Marine National Monument, so most people, when they think of marine monuments, they think of statues, or like someone told me they were worried about the monument that fell into the ocean when I was describing this. And it turns out there's actually more monuments than just statues. But under the Antiquities Act, it gives the President of the United States the ability to set lands aside for either historical preservation, cultural preservation, or for environmental preservation. And this area in particular, just like the other marine national monuments out in the Pacific, were set aside because of the unique structures, species that are found there, and biodiversity that's found there that we don't really have in so many places of the United States. Or we do, but preserving these places and giving them the protections so that they can maintain the current biodiversity, their structures, and giving future generations the ability to connect with these places as well. We're watching this right now, and it's amazing just looking back at technology and seeing how far advanced we've become, being able to capture this live and watch it live. But making sure that those places like these are in place for future generations to enjoy and also experience, whether it's through a telepresence or through mechanisms like Katie mentioned, like Mystic Aquarium, because these places are not easy to get to. It allows people to build that connection with the ocean, with the deep, and with those things that are usually not seen. One of the, oh, look at that guy. I'm gonna stop talking because I want someone to label that fish.
That's a chun acid. It's a little sea toad.
I think it's just beginning to come up in the live screen. There it is. Yeah.
Oh, and you can see its little Elysium that it uses. Oh, it might have just pooped.
It just pooped.
But on the front of its head, it's got a little lure that it uses for attracting prey.
That's so cool. You also see a lot of there was the poop. You see a lot of stuff floating around. We have a 7-year-old who wants to know, first of all, is this the midnight zone? And also, what's all that junk kind of floating around? Peter, maybe you're the best one. I'm not sure.
So I'm not quite sure. So this is well below mesophotic depths. I'm not sure what it's the midnight zone, and then it's always dark. So I guess the answer would be yes, at least in a general sense. Oh, there's the little guy again. These ought to be like a TV character, cartoon character. And the junk floating around is marine snow, which is, as we talked about, the bodies of plants, planktonic plants and animals that drift down after they die and turn into food for other organisms. We're far off the coast, so much of this is likely not of terrestrial origin. It's not sand and things like that. This is mostly organic material. Both again the drifting down bodies of plankton and organic material, dissolved organic material that comes out of solution and turns into aggregates. It's kind of like we see this if you stick your head in the water at the beach after a storm. A lot of the blobby looking things, sea snot in vernacular, is the organic material that comes out of solution. It coalesces on little bubbles from storms that churn up the ocean, churn up in the water. And these same kinds of processes happen as this stuff drifts down as well. So it looks like snow, but it's really the food that's driving the ecosystem down here.
So basically,
I'd like to add.
Go ahead.
I'd like to add that we are seeing a lot of this kind of stuff in the water column. So you see a lot of these dots that Peter identified as marine snow. So it's a lot of that organic material as well as sediments that are within the water column. But one thing that I want to point out is something that you don't see that's in the water column. So there's a lot of different kinds of chemistry that is involved in every layer. There's different layers of the water column that we call water masses. And sometimes they're brought on by currents that start off at the surface and then bow down and then enter into the deep ocean. And one of these things is over time, especially over geologic time, when I say that, I mean millions of years, we can start to develop that these kind of different elements in the water column can, what we use the term is precipitate. So they kind of settle on sometimes some of these rocks and some of these substrates that you're seeing here, and they actually can alter what we're actually seeing. And over millions of years in this specific area, we know that there are elements of what we call ferromanganese crust. So all this iron and manganese oxides that are within the water column are actually settling on the rocks themselves and can alter not only the outside of it, but can actually alter the interior structure of the rock as well. So sometimes it's really great to see, but as far as we're able to tell, it takes about 1 millimeter of this kind of layering on this rock can be up to 1 million to 2 million years worth of that encrusting on it. So it just goes to show how much time it can actually take for a lot of this to happen. So that's on the grand scale, that's a good side of things, but on the alternative, when it starts to leach into the actual original rock, it actually can remove what we would then use as age dating to determine what the original rock was and how old it potentially is as well.
Thank you, Kim. So we're seeing this super cool coral up close right now. Can you explain a little bit what this is, Peter? And also one of the other questions was, are these corals experiencing coral bleaching?
Well, so I'm looking at the scientific chat room where scientists from all over the globe are participating in this dive. I'm not sure if anyone mentioned that earlier. So this is really a group effort in terms of this kind of exploration with the Okeanos Explorer. And the coral that we're looking at now is an Acanthogorgia. It's a sea fan. And Scott France from University of Louisiana at Lafayette has done the identification in large part by the structure of the skeleton and the arrangement of the polyps to be able to do that visually. This looks like it might be certainly a meter and a half tall. This is a large colony for this species. So it might be. Actually, how tall is D2? Or how tall is the light bar off the seafloor from D2 on D2?
So D2 is 9 feet tall. So if it is about the same height as D2, it's approximately 3 meters or 9 feet.
Yeah. So they were just saying it's being shaded by the shades from the light bar, so it's tall. So this is a big tree. This is a great view as opposed to like being in Alvin where you got to crane your neck up at the low viewport to see something this tall. Coral bleaching. Coral bleaching is when corals extrude or evict the symbiotic algae that help create sugars and energy and help the coral grow. So it's a symbiotic relationship. That doesn't exist here in the deep ocean. There's no symbiotic algae that live within the coral, so that doesn't happen. It doesn't mean that the effects of climate change and ocean warming won't impact these species, and there's studies going on in many places around the globe on the effects of warming at different depths, as well as the effects on chemistry in the ocean on being able to deposit skeletons. The great thing about the monument is this can serve as a reference area to compare to the effects of warming and climate change elsewhere that also have the added effects of other human disturbances like fishing and mining and coastal development and ocean acidification, et cetera. Wow, that is just beautiful.
Yeah, you mentioned, Peter, I think this is a perfect example of why this area has been so pristine and why it's so important to keep it that way, because you have this incredible coral and clearly it's really fragile. And we talk about how these corals can be very long-lived, but very vulnerable to disturbance. Because I can see if it was capable of bringing a net or trawl or an anchor down in this area, how this thing could easily be crushed. So do you want to talk a little bit about that?
Sure. As Katie mentioned, most of the species that we're seeing, all of the species that we're seeing here are sensitive to disturbance, and many are vulnerable to human activities. And the issue is that we shouldn't do things in the ocean. We just might not want to do them everywhere in order to conserve the diversity that all these, to conserve the diversity, both species and genetic, that all these organisms contain, but also to be able to have places to be able to compare to others where we do things for human existence and for the utility of our economies, et cetera. And we are going deeper into the ocean to exploit natural resources. And having these kinds of protected areas is important for understanding what those impacts are and be able to balance the need for human uses and the need for conservation.
Thank you, Peter. So I think that explains why part of our exhibit is called Protecting Special Places in the Ocean, because this is a very special place. I have a question that is directed to Kim, and it's about the geology. So I think a lot of people are familiar with hydrothermal vents and how that supports these deep sea ecosystems. How does what we're looking at compare to hydrothermal vents?
That is an excellent question. So hydrothermal vents are sources in the deep sea that are actually really close to what we described before as like some of these hotspots. So really close to the Earth's interior where they're close to magma, so they can heat up really high. And this is usually a lot of organisms have, through evolutionary time, have adapted in order to be able to actually be near this really hot water. And essentially these hydrothermal vents are almost boiling water, if not boiling water, seeping through cracks and being able to actually then escape into the water column. But that does not describe necessarily the majority of the deep sea as we currently know it. Currently, the deep sea, the ranging temperatures are anywhere between 1 to about 3 or 4 degrees Celsius. So that means really relatively cold water we're looking at here. So in Fahrenheit, that means anywhere between 25 to, about 32 to about 38 degrees Fahrenheit. So really cold water that we're looking at here. And a lot of these corals are actually thriving in this environment. They've adapted to become these cold water corals. They're not just deep sea, but usually cold water as well. So some of these organisms, while some have adapted to these hydrothermal vents of extreme heat, a lot of these other organisms have adapted to this cold water too. And even though these seamounts were developed by that lava, by that hot spot that was able to then extrude all of this essentially magma that became this hard rock of volcanic material, which we call basalt, it's all cold now by now. So there's no heat being extruded from these seamounts. And you can see that currently we're actually taking a sample of this coral specimen that we've actually, we were eyeing before, this one that was just as tall as Deep Discover, which we call D2. And it's quite fascinating. Honestly, I'm a little bit distracted by it because it's so cool that we're
jump right to what's going on.
Yeah.
Talk about what's going on here. What kind of equipment is being used to do this, and how's this working?
Absolutely. So D2 is equipped with a manipulator arm. It actually has 2 manipulator arms, but we use one as the primary one, and it's a predator manipulator arm. And this is that claw that you're currently seeing coming into screen now. And it's equipped where our pilots back on the ship are currently have a like what looks like a replica of this manipulator arm that they're actually able to control with their arms and with their wrist and be able to control every hydraulic movement with this manipulator arm. And the claw itself is actually able to enclose, and it has a small little razor also built in, so we can actually only clip the exact section that we need or would want as a sample, so we can help preserve the rest of the specimen. So we're not collecting the entire specimen as a whole, and therefore it's allowed not only to continue thriving, but we also have a specimen that we can compare directly also to video footage that we're able to collect at the same time.
I think that really raises a good point, because I think we've just been talking about why it's important to conserve these places, and yet there are certain activities that are still allowed within the Northeast Monument, and one of them is research. So trying to better understand this living laboratory in hopes to better understand the ocean and how it will respond to things like climate change. But the other thing that is, you're also allowed to go out and do whale watching and recreational fishing. But I think it's important that people note that we're only taking as many samples as is necessary in order to really understand how this place works and all these really cool organisms. So thank you. Another question that a couple people asked, and I think this might be a good question for Casey, is where are we right now? Like, are we on the slope? Are we on the top of the seamount? Are we in the base of the seamount? Because I think everything's in this really close-up view from the ROV. It's hard to really understand where on the seamount we are.
Yeah, so we are targeting, so if you kind of I will not do it justice compared to the beautiful multi-beam bathymetries or the depth maps that we have from our mapping systems on board. I unfortunately can't just show you those data right now, but if you imagine the seamount is basically my hands. At the top of the seamount, that's where we're looking right now. You can kind of see how my hands maybe make tiny little peaks at the top. We're basically looking at a little peak at the top of the seamount. There's this tiny little feature that we've been systematically kind of climbing up throughout the course of the dive. So we started our dive today at just below 1900 meters. So I think 1940 meters, yeah. At approximately 1940 meters, so just over about 1.1 miles deep. And then we have been sort of working our way up slope. The good thing about dives like this is that we get to see lots of really incredible life. The bad thing is that we don't go very far because there's so much here to look at and so much here to see. We want to zoom in on everything. We want to take a look at it. We have such a global team of scientists that are participating with us that everyone wants to look at something different. So on these dives, we sometimes only go a couple hundred meters, unfortunately, but at the same time, it means that we get a really good look at everything that we do see. And we get to, like, the best dives are usually the dives that we go the least far because we're seeing so many things. So it's kind of a trade-off sometimes.
Unless you're interested.
But we're probably about, oh, go ahead.
I was just saying, unless you're interested in fish, of course.
We're like a mile deep, that far down?
Yep.
And so another question is, well, how cold is it down there? Like what's the temperature and the pressure like down so far deep? Obviously, like, we can't go down there, right? That's why we have to send robots.
Yeah, so a good way to, oh, go ahead, Peter.
Oh, no, go for it.
I was gonna say, to answer the pressure question, I've heard it best described to me when I'm trying to understand pressure units, which is a pretty esoteric thing that in my brain doesn't always make sense. But essentially, if you took an elephant and had it stand on your pinky toe, that's the kind of pressure that you would be experiencing at depth. So it's a lot of pressure down there. So human beings cannot actually withstand this pressure. That's why we use remotely operated vehicles and these robots to go down and take a look for us. We get to sit up on the nice cozy ship or be at home and don't have to worry about those elements ourselves. Peter, were you going to jump in? Sorry about that.
No, that's fine. So the temperature is 3.6 degrees Celsius, which is 38.5 degrees Fahrenheit, which is chilly. And I mean, that's a good temperature. Yeah, exactly. And that's a good, excuse the background noise. There's big construction trucks going by my house. And that's a good analogy for pressure. If you had, like a lung full of air, or on the outside of the housings that contain all the electronics of the ROV, if you're a fish and you're essentially made up of all water, you don't sense that pressure. At least at these shallower depths. If you're at 6,000 meters, there are some species have adaptations to both the cold and the pressure. But it would be, it's much like scuba diving, where the pressure is ambient. You're breathing air at the same pressure that the surrounding water is. So you really don't feel that pressure per se. But send down a styrofoam cup, and it turns into a little shot glass.
So last question, because we've had such a great audience, and this is from Everett, who's 6 years old. Can sharks go this deep?
Oh, absolutely. So there's a diversity of deep sea sharks that are adapted to the cold, to eating very infrequently.
And I know in our exhibit, we have some video of a Greenland shark, which is also rare in the monument. So we are unfortunately out of time. It's been a fabulous audience. We've had over 200 people tune into this. Tune into the rest of the expedition on the Ocean Explorer website. Come to Mystic Aquarium to see our new exhibit. And I'm just going to turn it over, thank our panelists, and turn it over to Brittany to close us out.
Yeah, I would like to thank all of our panelists for coming in today virtually and joining as we get to explore Retriever Seamount, which really truly is an incredible view and so much diversity. And as was mentioned earlier, doing this type of research, this type of collection is going to help us better understand the management needs for this marine monument, especially being the first one in the Atlantic. So we know what it needs, what we can provide it, and give it the voice that it so, obviously deserves, with its beauty and structures and unique creatures that are all in there. So I appreciate everyone tuning in, and follow the Ocean Exploration online, Facebook. They will be, as mentioned earlier, they will be diving in the Southeast later this year. And we look forward to finding other ways to bring the monument to you, because this is your monument. This is your marine national monument, just like your just like your national wildlife refuge. And we're glad you're here and getting to experience it with us.